and how this tumor cells are going to be, whether it's a benign or a malignant tumor. And if it is a benign tumor, what is its properties? And if it's a malignant tumor, what are its properties, and what are those genes that are responsible for tumor cells, which leads to malignancy. So if you can just see this picture, you can see the differentiation of how a normal cell looks like and how a cancerous cell looks like. There's a clear difference in the morphology of the cells. The normal cells are a bit smaller, whereas in the cancer cells, the cells are going to grow a little bit bigger. Now, what do you mean by cancer? It's just an umbrella term for a large group of diseases caused when abnormal cells divide rapidly. Because cancer means it can be of any type of cancer. Say, for example, if it's a brain cancer, a breast cancer, lung cancer. Cancer means you will know that it is some type of abnormal cells, but where it is originating is a name that is given, whether it is if in the brain, it's called as a brain cancer. If it's in the lung, it's called as a lung cancer. So it's an umbrella term, a complete single name for different diseases. So the name cancer over there, which spreads to other tissue and organs. It is one of the leading causes of death in the world. Now, if you can check in the pic over here, the picture shows how a normal cells looks like and how a cancer cell looks like. Okay, so in the normal cells, if you can see, there is a large cytoplasm. So this is the cytoplasm, the nucleus over there, and then the nucleolus inside the nucleus, and then the chromatin, that is the chromosomes, how they are present over there. So it has a large cytoplasm around, and there's a single nucleus with a single nucleolus present in them and a fine chromatin present. But when you see the cancer cells, the cytoplasm becomes shortened. If you can see the difference over here, the normal cell cytoplasm and the cancer cell cytoplasm. The cytoplasm is a little bit shortened with numerous nucleus present. That is a multiple nuclei. Here, usually in a cell, you should have a single nucleus, but here you have a double nucleus over there. And the multiple large nucleolus, if you can see in one cell over here, you have 3 nucleolus over there. So there's excess of nucleolus present over there, and you can also see the chromatin being a little bit differentiated over there. So granular chromatin is what you can see over there. So this is how the normal cell can change into a cancerous cell over there. Okay, so what do you mean by cancer cells? That's how and where it grows. We are made up of trillions of cells. Now, the cells have to grow and divide as a body needs them to function daily. That is, we tend to lose a number of RBCs daily, but again, we tend to gain number of RBCs. We tend to lose number of cells that are aged due to apoptosis, the cell dies. But again, your cell have to be dividing so that it compensates the death over there. So the body has to divide into numerous cells so that you are compensating with the dead ones over there. The healthy cells have a specific life cycle. If you remember the cell cycle, the GO, G1, S phase and G2 phase over there, and then the mitosis phase, it has a specific life cycle, reproducing and dying simultaneously. And the new cell takes up its place over there. Now, what happens during cancer? The cancer disrupts this process and leads to abnormal growth in the cells. That is, when you have a wound, say for example, and your cells are going to divide over there, and it is going to repair the wound over there. But after the wound has been repaired, the cells are given a signal to stop dividing because the wound has been healed. If the cells are not going to listen to the information that is given to them to stop dividing, it can become abnormal growth over there, and this abnormal growth in the cell leads to cancer, and this can be due to mutations in the DNA. Now, when this mutations are not corrected, then that can become cancerous cell. So mutation can cause cells to survive instead of cell death. So that is what it's meaning of your mutations. Because once the cell has been healed up, when the tissue is healed up, the cell has to stop dividing. But due to mutations, what is happening? The cells, the old cells, instead of dying, they are still surviving. And this also produces again and again. Then there is a numerous number of growth that is being formed that leads to cancerous cell due to the cells dividing uncontrollably. Now, tumors are clusters of cells over there, and yeah, you will have to talk that over there. And, So these are clusters of abnormal cells. If you can see this picture over here, so these are an abnormal number of cells. So you have a single cell over there. The single cell divides again and again and becomes an abnormal cell. That is, they swell. If you can see in the picture, they are swelled over there. And the names of the different types of tumors are suffixed as OMA. That is, in the last end, wherever you see an OMA over there, then you can say that it is a tumor cell. So usually the cells have to divide and proliferate under tightly controlled conditions called balanced program cell death. That is, once the cell has completely completed its work, then they have to die because of aging. So that programmed cell death is called apoptosis. So you will be talking about apoptosis and necrosis. Necrosis, they are dying without any, that is, apoptosis is programmed. So only that cell is going to die. But during a necrosis, the cell is dying. Along with that, it is also killing other cells around them. That is necrosis. But here, programmed cell death will lead to a normal cell growth and cell death over there. But however, mutations which occurs in the cellular DNA will affect this balance. That leads to stoppage of cell death, and uncontrolled division leads to this number of cells over there, which is going to grow in n number, which swells in size. So tumors can vary widely in size and also develop almost anywhere in the body. And some cancer cell can also migrate through the bloodstream or lymphatic systems to distant areas. And this is called metastasis. Now that is, when the cancer has occurred, it is going to occur in one part of place. Say, for example, it has occurred in the lung. But after the tumor has been formed over there, which has become malignant, that is cancerous tissue. Now, this will have to need more proteins or more food to grow. So what they do is they divide over there, they chop off from themselves from there, cut off themselves from there, and they go into the bloodstream. From the bloodstream, they go and search for a different place to survive over there. That's called metastasis. That is, moving from one place to another place is called metastasis of the cancer cells. So people often associate tumor with cancer. So for example, you must have heard some people saying that they are suffering from tumor cells. So the moment you hear tumor, you shouldn't get confused with a benign tumor and a malignant tumor. So you have to first confirm whether that tumor is a benign tumor or a malignant tumor, because benign tumor is not going to form a cancer, but a malignant tumor can definitely become a cancerous. So benign tumor is non-cancerous, whereas malignant tumor is a cancerous. So you will have to first understand, whenever some people or doctor says that a person is suffering from tumor, you are going to make sure that whether he is suffering from a benign tumor or a malignant tumor. So coming to types of cancer, the cancer are named from the area in which they begin. That's what I told you all. And they spread to other parts of the body. But the name is given to that cancer where it has been first identified, from where it has originated. That is the name that has been given. So for example, if you find them in the breast, then you're calling them as breast cancer. If you're finding them in the lungs, then you're going to call them as lung cancer. And if you're finding it in the liver, it's called as liver cancer. So there are several clinical terms used for certain general types of cancer. That is, a carcinoma is a cancer that starts with the skin, lungs, breast, pancreas, and other organs and glands. So when you're talking about carcinoma, it starts from, it can occur in the skin, it can occur in the lungs, breast, pancreas, and other organs and glands. So then you call it as carcinoma. When do you call it as sarcoma? Remember, OMA means we are going to talk about cancerous cells. Sarcoma is a cancer of connective tissues such as the bones, muscles, cartilage, and blood vessels. So wherever the connective tissues are present, and if you find any cancerous cell in that connective tissue, then it is termed as sarcoma. So whenever you hear a sarcoma, then you have to think that there's a cancer in any connective tissue, that it can be a bone, muscles, cartilage, or blood vessels. Leukemia, of course, blood cancer is a cancer of the bone marrow where the RBCs are going to develop, that is hematopoietic stem cells, which creates the blood cells. Lymphoma are cancers of lymphocytes, that is the lymph nodes, and melanoma arises from the skin. That is a pigment of the skin, the melanin pigment. So if there's any problem in the skin, then you have the skin cancers over here. Those are called the cancers of the immune system. Now, you can see in this picture over there, that's the cancer in the lungs over there. So you have a lung cancer over there. Then you have the melanoma on the patient's skin. So this is because of the cancer that is present on the skin due to the melanin pigment. And that's called as melanoma. And any soft tissue sarcomas, you can find in the lymphatic vessels, in the blood vessels, or in the fat, or in the muscles, bones, ligaments, and tendons, or also in the nerves. So these are the different types of cancers, that is carcinoma, sarcoma, lymphoma, and melanoma. Now, characterization of benign and malignant tumors. Now we'll talk about how do you differentiate between a benign tumor and a malignant tumor. So if they ask you a question to characterize benign and malignant tumors, you're going to talk about this. So if you can see in this picture over there, the benign tumor, there is a normal cell, and in between there's a tumor cell. The tumor cell has not metastasized. It is placed over there and they have not moved over there. And it is properly having a boundary wall around them. Whereas in a malignant tumor, you can see the tumor is spreading. It is not going to stay in one place. They are going to move from one place to another place. So that is a malignant tumor. So we'll be talking about all this in a short while. Okay, so what do you mean by malignant tumors? If you can see in this picture over there. So these they stay at a primary location without invading the other parts of the body. They do not spread to local structures or to distant parts because they are going to stay at one particular point. The tumors are not usually problematic because you're talking about benign over there. So they are not at all problematic. And however, they can become large and compressed structures. If you can see in the picture over here, the lungs tumors that is present over here, it's going to compress the trachea beside them. So there's a lung cancer which is pressing the trachea over there. So when it is pressing something over there, that is for example, a trachea or a windpipe, the windpipe, when it is pressed, you feel a difficulty in breathing. But otherwise, if it would have been a little bit further from that lungs over there, then it wouldn't have made any problem. So this would warrant an urgent surgical removal. Benign tumors are unlikely to recur once removed. So once it is removed, you won't be having any more problem in the future because the benign tumors are not going to grow because you have completely removed those cells. So that is why it's called as benign tumor causes. It is not at all a cancerous tumor because we are not going to call them as malignant. So simply you can remove it. So you don't, that is what I'm telling you. You are not going to get tensed up when you hear the word tumor because you have to understand whether it is a benign tumor or a malignant tumor. So here we are going to talk about benign tumors. First thing what I said is they're going to stay at the primary location. They're not going to move from that place, and they have a proper boundary wall which does not allow them to move anywhere from its point of origin. Only when it is problematic, then it has to be removed. Otherwise, there is no problem at all. Sometimes when the doctor says, yes, it has to be removed, then you have to go for surgical, but you don't have to worry that it is going to be a malignant one. So what are the different types of benign tumors over here? First one, you can call it as adenomas. So what is this adenomas? The adenomas are polyp-like structures. If you can see in this picture, there's a polyp-like structure in the large intestine. Usually they develop in the gland-like cell in the epithelial tissue, the glands, which secretes any mucus over there. So they are going to form these polyps in a gland-like cells in the epithelium, a thin layer of tissue covering the glands, organs, and other structures. So the treatment depends on the location and the size, and some colon polyps are adenomas and should be removed in case they become malignant over here. So here you can see there are some polyps. There might be some numerous polyps, aggressive polyps. Here you're going to see only 2 polyps. You can be having more number of polyps over there. Until and unless it is going to pose a problem in the humans, you don't have to worry about it. But then if it is going to be a problematic, then the doctor is going to definitely surgically remove this polyps. Again, this is a benign tumor in the large intestine. Sometimes it can become malignant. Fibroids, you must have heard of this word fibroids in the uterus, because every now and then we keep hearing any woman having fibroids in the uterus. So these are tumors of mesenchymes. Fibroids grow in the fibrous tissues over there. Uterine fibroids are common, affecting 20 to 80 of women by age of 50. So that is the reason why mostly you nowadays keep hearing this word fibroids in the uterus, because this fibroids, because there are so many problems in that it can be due to the environmental condition, it can be due to the food that we eat over there, which is having more number of carcinogenic agents. Say, for example, if you're going to eat a lot of preservative foods or packaged foods, or the beverages that we drink nowadays today, which has lots of pesticides and artificial coloring agents. So all these are going to create all these different problems over there. So this fibroid, sometimes here we are going to talk about benign tumors. So benign tumors is something, a fibroid, and they don't necessarily need any treatment. So you must have seen some doctors saying that this can be cured by medicine. So there is no problem. But sometimes if the size has been increasing, and if that the size is going to create a problem in the uterus, if you have a constant stomach pain or something like that, then definitely this has to be removed surgically. Otherwise, there is no problem with this fibroids over there. Okay, there's one more called hemangiomas. Hemangiomas is of the blood vessels over there. It's a type of tumor that's made up of extra blood vessels. That's why hemangiomas, hemoglobin, hemangiomas, problem in the blood vessels. They are the most common tumors in the children. They tend to occur more often on the skins and the liver. So if you can see in this picture, the baby is suffering from this hemangiomas. Sometimes you call it as a birthmark over there, because when the child is born, immediately you can see all this patches of red patches on the body of the children over there, usually on the skin and the liver. On the skin, if this present on the skin, they may initially appear as a red birthmark over there. Then over the time, it will start to form a red lump, very small lumps. Although they should be monitored, hemangiomas typically don't cause any problem and usually fade away without the treatment. So you must have seen in some children, you must have seen a red patch on the skin, on the back or on the hands, on the thighs. You must have seen some red patch. Initially, it might be very dark, but usually it can fade away. But otherwise, if it is going to form into a lump, then you will have to monitor for the growth, so that you can be sure that this benign doesn't turn into a malignant one. Then lipomas. See, the lipomas means fatty tissues. So there is a slow growing tumors that form fatty tissues under the skin, and they can occur anywhere, but particularly in the neck, shoulders, armpits, and trunks, if you can see in this picture. And mostly these are common between the ages of 40 and 60, and treatment isn't always necessary, but sometimes if it is going to bother, then it can be removed surgically. Now, malignant tumors. Now we are going to talk about malignant tumors. Till now we were talking about benign tumors. So you're not going to worry about benign tumors because they are not going to cause any cancer. But when you're talking about malignant tumors, then definitely these are cancerous cells which can pose a problem in the individual who is affected with this malignant tumor. So what is the characters of this malignant tumor? The malignant tumors are cancerous with a capacity to spread and establish new tumors in other organs or tissues in the process called as metastasis. So they are going to move from one place to another place. If you can see in this picture over there, the malignant cancer, it has formed in one place, but they are going to metastasize. That is, they are going to move from its origin to other parts of the body, because they want to spread as much as possible, because they want more amount of blood supply to them. So they are going to metastasize, or they are going to move from one place to another place from the circulation, and it can reach to any part of the body in from the place of origin. That is called as metastasis. So whenever there is a malignant tumor being formed, it is always better to find out if it is in an initial stage. As we all know that the cancer is made up of 4 stages: stage 1, 2, 3, and 4. If you're going to identify this cancer in stage 1, then you can control it, because it has not metastasized or it has not moved from that place. So it is easy for the doctor to just remove it and give some treatment to control it. But once it is metastasized, then it will become a little bit difficult for the treatment to process. So during metastasis, primary malignant tumor spreads to secondary sites, and cancer cell breaks away from the tumor and spread to different parts via the blood or lymph system, and they metastasize to liver, lungs, brain, or bones. So that becomes very dangerous over there. And what is the treatment? Malignant tumor should be quickly removed surgically or by chemotherapy or radiotherapy. Sometimes both combination is needed in severe conditions. So there are several established characteristics of hallmarks of cancer, such as inducing the production of new blood vessels, that is called angiogenesis, and sustaining growth promoting cell signaling. So that's what we are talking about. When the cancer has to develop and when the cancer has to survive, it needs blood supply. Now, when it is needing blood supply, what it does, it cuts off the connection to the normal cells. You have capillaries going to the normal cells. Now, what it does is it cuts off the supply to the normal cell, and it makes sure that the angiogenesis is developed in its cancer cell, towards the cancer cell. That is called angiogenesis. So first is production of new blood vessels for them so that they can survive with the blood supply. Second is sustenance of growth promoting cell signaling. So cell cycle, if you remember the cell cycle we are talking about, it needs a continuous mitogenic activity in those cells. Now, once the mitogenic activity is continuously progressing, that means the cells are going to divide n number of times, and then that is where they are going to grow into cancerous cell. So they need angiogenesis and growth promoting cell for cell signaling for the development. So there are many types of malignant tumors. We'll be talking about one by one. First is carcinoma. The carcinoma develops in the epithelial cells, and the most common type of the cancerous tumors, representing 80 to 90% of the cancer. So wherever you find or hear cancer, 90% of the cancer is of carcinoma, wherein the examples include adenocarcinoma forms. These are produced in the fluids and the mucus. Remember the epithelial cells inside. This includes breast cancer, colon cancer, and prostate cancer. So these are the places where the fluids are produced and the mucus also being formed, so that it is taking care of the colon and the prostate. In the breast cancer, for the because these cells are going to produce milk, that is the reason why they are calling them as glands, where they are going to produce fluids and mucus. So adenocarcinoma means the cancers in the breast, colon, and prostate cancers is called as adenocarcinoma. Basal cell carcinoma starts at the lowest layer of the epidermis. If you remember the epidermis structure, the lowest layer is the basal cell carcinoma, from where the cancer cell can grow. And this is how you can see in this picture over there, the basal cell carcinoma. Then squamous cell carcinomas, the squamous cells that you find, the shape, if you remember the histology of the cells, you're going to talk about the squamous cells. These are cells that is just beneath the outer surface of the skin, as well as the organs like bladder, intestine, kidney, or stomach. You find this squamous cell carcinoma forming. And transitional cell carcinoma. So what do you mean by transitional cell carcinoma? These are the tissues develops in the tissue called the epithelium cells, called urothelium. Definitely it's in the excretory cells, that is in the bladder, kidney, and the ureter's cancer may be of this type. So that is called as transitional cell carcinoma. So that is examples in the carcinoma: adenocarcinoma, basal cell carcinoma, squamous cell carcinoma, and transitional cell carcinoma. So the next one is a sarcoma. So the sarcoma over here, remember we were talking about the connective tissue disorders in a benign tumor. That benign tumor can form cancerous. So when it's forming cancerous, then you're calling us as different types of carcinomas, the sarcomas, which develop in the bone, cartilage, fat, muscle, tendons, and ligaments. If you can see in this picture, the examples includes osteosarcoma, that is a tumor in the bone, is called as osteosarcoma. Chondrosarcoma is the cartilaginous cancer. So that's the chondrosarcoma, that is a cartilage over here at the kneecap over there. So the cancer in the cartilaginous bones. Liposarcoma, that is at the fat, wherever there is fat present. if you find those cancers over there, then that's called as a liposarcoma. And rhabdomyosarcoma, this is the cancer in the muscle cells. Another name for this rhabdomyosarcoma is leiomyosarcoma. So that is the leiomyosarcoma in the muscles. If they are present in the muscles, then that's called as leiomyosarcoma or rhabdomyosarcoma. So if you hear the name sarcoma, then you're talking about connective tissue cancers. Then the next one is blastoma. Blast, that is the immature cells. Remember, blast, clast, and sites. So immature cells are called the blast cells. Any cancer that is produced in the immature precursor cells, then it's called as blastomas. These malignant tumors are common in children because in children you have this new immature cells forming a lot. So mostly you find this type of cancer in the children, and because these are also thought that it can occur due to some genetic error in the fetal cell development. And the examples include glioblastoma. Glio is a glial cells, if you remember, the astrocytes that supports the nerve cells within the brain and the spinal cord. So if there's a problem in the nerve cells, then glioblastoma. Retinoblastoma, the blastoma that you find in the retina, wherein the complete retina is spoiled over there for the cancer cells being formed in the retina over there. So that's called as retinoblastoma, the light sensitive cells of the retina in the back of the eyes. Hepatoblastoma, cancers occurring in the liver cells are called as hepatic cells. Hepatic means liver. So hepatoblastoma is cancer occurring in the liver cells. Mostly it is on the right lobe of the liver, but sometimes conditions can differ, and you can also find it on the left side of the lobe of the liver. So that is blastoma cancer cells. So these are the two different types we talked about of benign tumor cells and malignant tumor cells. I hope you have no idea. You have no problem in understanding this benign tumor cells and malignant tumor cells. If you have any doubt, please ask me again. Okay. We'll go to the next aspect of the malignancy, that is molecular aspects of transformation and malignancy. So you're going to talk about the molecular aspects of malignancy. So what is it? So if you can see in the cells over there in this picture, you have a normal cell, and if this normal cells undergoes mutations, the first mutation, nothing happens over there. But if this is carried forward in the second mutation, it is going to grow in number. And if it is going to carry forward in the third and the fourth mutations, then this mutation is going to lead to malignant cells. Why? Because the cells have lost its the proteins that is going to be giving it the signal pathway to start cell cycle and to stop cell cycle. Here, what is happening is this normal cells, once it's undergo mutation, it has stopped its normal cell signaling, and it is going into excessive cell division growth over there, and that leads to malignant cancer. That is, it all starts with mutations or different problems over there. So what are the different problems you're going to talk about over here? So what are the genes responsible for tumorogenic cell growth? So first thing is, you are going to talk about proto-oncogenes. So for example, in the normal cells, you have proto-oncogene cells, which leads to cell growth and proliferation, first oncogenes. Tumor suppressor genes, that is, these are the genes that suppresses a tumor from growing. So you find these two genes in the normal cells, proto-oncogenes and tumor suppressor genes. So just make a note of it. What are the genes responsible for tumorogenic cell growths? So we are going to talk a lot about this proto-oncogene and tumor suppressor genes. Now, what is happening in the cancer cell? So in the cancer cell, this proto-oncogene is mutated or it is highly activated, which becomes oncogenes. So proto is removed, and then it becomes oncogenes. And once this proto-oncogene is activated, it turns into oncogene. That leads to malignant transformation, if you can see in this picture over there. And when there is a loss or mutation occurring in the tumor suppressor genes, so in case you lose the tumor suppressor genes due to any varied region, or if there is a mutation in the tumor suppressor gene, that also leads to malignant transformation. So we are going to talk a lot about proto-oncogenes and tumor suppressor genes in this coming slides. So how they are going to transform, that is called malignant transformation from normal cells, how it is transforming itself into malignancy is what we are going to talk about over here. So here the normal cells acquires the properties of cancer because of transformation, and this occurs by a series of steps, and the affected tissue gradually accumulates the genetic mutations. You can see in this picture. So they're going to gradually get the genetic mutations over there, and that leads to malignant phenotype. This damage can be a result of endogenous processes inside, such as error in replication, intrinsic chemical instability of the cell, certain DNA bases, or from the attack of radicals generated due to metabolism. Free radicals generated due to during metabolism. So here you can see if there's an error during replication. So here you are going to see original DNA. Now what is happening in the original DNA? This G is connected with the C, that is guanine to cytosine. But due to error in the replication, the guanine is going to get attached to the thymine, which is not at all a pair, because thymine has to attach to adenine, a guanine to cytosine. But there is an error in the replication. So what happens is this, when it is being copied, one side of the DNA thread is going to copy the correct one, whereas the other side, it is going to form a mutant cell, because that is not an original pair that has to be present over there. So that gives a wrong protein signals. The codon over here has been changed from CT G to CTA. That gives a different type of proteins over there. So that leads to cancerous development. Then another type is chemical instability of certain DNA bases, and then from the attack of free radicals generated during metabolism. Now, what do you mean by free radicals generated during metabolism? Free radicals are nothing but, for example, if you're going to talk about free radicals, you can talk about superoxides, the oxygen, which is having one incomplete electrons over there, unstable electrons over there. Then you have nitric oxides, that is also one more free radical. And you also have a hydrogen peroxide, H2O2. So these three, and there are some other free radicals also present, but these three have to be controlled. The superoxides are removed by the enzyme called superoxide dismutase. The nitric oxides and the other superoxides are removed by the you have catalases, you have superoxide dismutases. There are different types of enzymes which tries to control them. But if there is a problem in that enzyme, so for example, if there is a problem in the superoxide dismutase, now this is not going to metabolize the superoxide. The accumulation of the superoxide increases the hydrogen peroxide in our body, and all this accumulation leads to cancerous cell, because the metabolism is not occurring smoothly, and if there is any problem in the metabolism, that cell is going to derail itself, and then they are going to lead to cancerous growth. So this DNA damage can be a result of interaction with exogenous agents also. We have talked about endogenous, that is error in replication and free radicals production. But not only endogenous process, due to exogenous agents also, outside agents also can create cancer. That is, first one is due to radiation. There are a lot of different types of radiation that you might come across. Then you have UV radiations and chemical carcinogens. UV radiation is, that is when you're going to get yourself exposed to too much of sun rays, that is the mid afternoons between say 1 to 3 o'clock. If you're going to get yourself exposed too much of that sun, then you're exposing your skin to too much of UV radiation. That is where we are coming up with a lot of creams over there, wherein the skin is going to be protected by UV radiations over there. So UV radiations one, and chemical carcinogens. So what I was talking about, chemical carcinogens, that is in the food items nowadays. Nowadays you are coming across a lot of inspection going on in different hotels over there. These hotels are also having a lot of food coloring agent. All these food coloring agents can have this carcinogens effect. Then the ready-made food products for preparation, they all have preservatives in them. That preservatives also have this carcinogens. So try to minimize the usage of this coloring agents and ready-made food items, wherein you are just going to bring them, heat them, and all those. These are also going to create carcinogens. So these are UV radiations, and chemical carcinogens are two important ones for exogenous agents over there. And the cells usually have the ability to repair such damage, but for various reasons, errors can occur, leading to permanent change in the genome. Mutations are introduced. So if there's a permanent change in the genome, say for example in this picture over there, if there's a permanent change in the genome, then you're going to have a different types of proteins which will reduce the cell repair and leads to mutations. So that is the reason why during cell cycle you have a phase called G1 and G2. Now G1 and G2 are the checkpoints where the DNA is going to repair itself. You're giving time, the cells is going to give itself time to repair itself wherever there's any DNA repair that has to be needed. But during mutations or due to any different changes in the program or signaling cell, there you have a problem, and this does not allow checkpoint to occur, and that leads to mutations. Now, what do you mean by a proto-oncogene? We are talking about proto-oncogene and suppressor, tumor suppressor gene. So we are going to first talk about what is a proto-oncogene. The proto-oncogene is a normal gene that could become an oncogene due to mutations or increased expression. So we all have proto-oncogene. The presence of a proto-oncogene will create a normal gene in us, but due to various changes in the cells, that can lead to increased expression of oncogene. The proto-oncogene codes for proteins that helps to regulate the cell growth and differentiation, and these are involved in signal transduction and execution of mitogenic signals. Remember what is mitogenic signal? Mitosis. So upon acquiring an activating mutation, a proto-oncogene becomes a tumor-inducing agent called as oncogene. The proto is removed and that becomes onco, and this oncogene leads to different types of cancer cells. So the examples of the proto-oncogene includes the RAS. RAS is a rat sarcoma. The full form is rat sarcoma that was first found in the rats. So the name RAS. You also have some different RASes in humans also. We'll be talking about that. Then you have WNT, WNT proto-oncogene, MYC oncogene, ERK, and TRK. The MYC gene is implicated in Burkitt's lymphoma, that is lymphoma in the lymph nodes is called as a Burkitt's lymphoma. Another example of an oncogene is a BCR-ABL gene found on the Philadelphia chromosome. Remember when we're talking about the syndromes over there, the chromosomal abnormalities, there we were talking about Philadelphia chromosome. That is the translocation between chromosome number 9 and 12 leads to myelogenous leukemia. The BRCL-ABL code for tyrosine kinase, which is constitutively active, leading to uncontrolled cell proliferation. So BCR-ABL is a gene found on Philadelphia chromosome. That is when the translocation occurs, this is activated, BCR-ABL is activated, that leads to uncontrolled cell proliferation leading to cancers. That is what cancer? Myelogenous leukemia. So here you can see all the different types of cells over there. The growth factors, EGF, can lead to cancer. Growth factor receptors, ERB, can lead to cancer. Then you have RAS leading to cancer, RAF, and all these activates MEC genes, and the MEC genes leads to ERK, and the ERK is also having a FOS gene. There's another kind of a gene that encodes potentially for oncogene, and all these leads to cancer cells. The proteins with known oncogenic potential, these are all highlighted with yellow. So wherever you have this highlighted yellow one, all these are going to form cancer cells in the plasma membrane or in the cytoplasm or in the nucleus over there. The oncoprotein function. So oncogene, proto-oncogene to oncogene. Oncogene is an oncogene protein functions. What are they? The oncoproteins are actually vital to human survival. Thousands of them in our body ensure that the cells grow and divide, and they help heal wound, repair genetic damage, and boost our immune system. Which one? Oncoproteins. But when oncoproteins stop working properly, they promote transformation of cells in the tumors by dysregulating the signaling pathway that are involved in the cell growth, proliferation, and cell death. Cell growth has to occur, mutations has to occur, mitosis has to occur. And once the mitosis occurs, you're going to proliferate the cells. After the cell has been proliferated, after the work is done, they have to die. But here, there are no death occurring over there. That leads to cancer cells. The resultant protein encoded by an oncogene is termed as oncoprotein, and it plays an important role in regulation of synthesis of protein linked to tumorogenic cell growth. And some oncoproteins are accepted as tumor markers. So if you want to find out if a person is suffering from different types of tumors, then you have this tumor markers, which is called as polypeptide growth factors, growth factor receptors, elements of intracellular signaling pathways, and transcription pathways. So all these proteins become a tumor markers. If you get a positive result from that, then definitely we can say that a person is suffering from any type of cancer over there. Okay, so what are the different types of oncogenes, and what are the cell functions or gene function it gives us? Growth factors or mitogen, the function is it induces cell proliferation. Examples of here is CCIS, and if there's a problem in them, that leads to glioblastoma, osteosarcomas, or breast carcinomas, or melanomas. When you're talking about receptor tyrosine kinases, their function is to transduce signals, growth, and differentiation. What are the examples of here? EGFRs, PDGFRs, and vascular endothelial growth factors. That leads to, if there's a problem over here, that leads to breast cancer, gastrointestinal stroma, gastrointestinal stromal tumors, and pancreatic cancer. The example over here, cytoplasmic tyrosine kinase, cytoplasmic, present in the cytoplasma, tyrosine kinase. These are again receptors for proliferation and differentiation. Example, SRC family, SYK, SAP70 family, BTK family, ABL gene in CMN, that is Philadelphia syndrome that you're talking about. That leads to different types of colorectal or gastric or pancreatic cancers. The other one is a transcription factors, where transcription of the protein has to occur. Example is the MYC gene over here. The MYC gene is a myelocytomatosis gene over here. Myelocytomatosis gene regulates transcription that induces proliferation. Cancers are malignant T cell, lymphomas, acute leukemias, or breast cancers are present over there. Transcriptional cofactors. So you also have a cofactors, YAP and WWTR1 genes leads to glioma, melanoma, lung cancer, and breast cancers. Now, cytoplasmic oncoprotein. So you're going to talk about two types of oncoproteins. One is cytoplasmic oncoproteins and the nucleoplasmic oncoproteins. You might be asked in the exam to discuss cytoplasmic oncoproteins and nucleus oncoproteins. So this is what you're going to write when you're going to talk about cytoplasmic oncoproteins. So cytoplasmic oncoprotein is a serine threonine protein kinase. Underline that word, serine threonine protein kinase, which plays a role in regulation of cell proliferation, programmed cell death, and cell differentiation, and of course, embryonic development. So this protein is very, very important for programmed cell death, that is apoptosis, proliferation, and embryonic development. So this is present where? It is present in the cytoplasma of the cell, the cytoplasmic oncoprotein. And this is the examples of the cytoplasmic oncoprotein are RAS, remember, rat sarcoma, PIM1, MOS, and COT genes. So these are the genes that are present in the cytoplasma. About a third of the human cancers, including a high percentage of pancreatic, lung, and colorectal cancers, are driven by mutation in the RAS genes. RAS genes leads to different types of diseases or cancers over there. The main members of RAS gene families are, these are found only in the humans. Which one over here? KRAS, HRAS, and LRAS genes, which encode proteins that have an important role in cell signaling, that is cell proliferation and embryonic development. When the RAS genes are mutated, cell grows uncontrollably and evades cell death, leading to cancers. The RAS mutation also makes cell resistant to most available cancer therapies. So if you have a problem in the RAS protein, then whatever therapies you want to give them, the therapy is not working. Why? Because there is a mutation in the RAS gene family. Now, how is this working? How this RAS works? If you can see in this picture over here, RAS gene with GDP inactive, but when this RAS gene has to be activated, it needs to have the GTPs in them. So once the GTP is there, it leads to controlled growth, proliferation, and migration. Now, once the function is over, then the GTP is formed into GDP, triphosphate becomes diphosphate, guanine, triphosphate to guanine, diphosphate. So when it goes to GDP, that means the cell is inactive. That is, the cell is functioning normally. But what happens during mutation? During mutation, the RAS is stuck in the active state of the GTP over there. That is, the GTP is not becoming GDP. It ignores the cell signal and drives to cancerous cells. So here, when the RAS is working thoroughly, that means the RAS is interchanging itself from GTP to GDP. When it has to proliferate, the RAS becomes active by GTP. Once the work is done, embryonic development is done, it has to stop over there, and it goes to inactivated state, that is GDP. But in the cancer, because there's a mutation in the RAS, the mutation gets stuck in the GTP state, and they do not allow the cell death to occur that leads to cancerous cell. That is cytoplasmic oncoprotein RAS genes, RAS, rat sarcoma. But this name was because the name RAS is given because it was first identified in the rat, so rat sarcoma. But those cells that are found in the humans are called as KRAS, HRAS, and NRAS. Then you have another type of genes over there, NF1 gene in the cytoplasm. That's the neurofibromatosis 1. NF is neurofibromatosis 1. This codes for RAS GTPase, RAS GTPase, activating proteins that causes neurofibromatosis type 1. And they can contribute to disease like non-obese diabetes and diabetic retinopathy. Non-obese is, even if you're not obese, you have diabetes. So that is because of the neurofibromatosis 1 gene, which codes for RAS GTPase. And also, it also leads to diabetic retinopathy, problem in the eyes. So once you have diabetes, then you have different types of diseases occurring. Now, what happens during this activation? Specific mutations occurs at codons 12, 13, or 61 in the RAS gene, which is associated with tumor cells. Codons. Remember the codon, the 3 base pairs, which gives you 1 codon. That codon is the one that gives you a specific proteins. So here, the RAS activation is because of this neurofibromatosis type 1, which gives GTPase activations. So there is a specific mutation, maybe at codon number 12, 13, or 61, which gives activates the RAS for tumor formation. Then pancreatic adenocarcinomas are some of the most frequent detected tumors because the mutations are in KRAS at codon number 12. Mutations at codon number 12 and 13 leads to colorectal carcinoma. Then you have one more gene called PIM1 gene, expressed primarily in the B lymphoid and myeloid cell lines. Remember, B cells and the myeloid cell lines. It leads to signal transductions in the blood cells, contributing to both cell proliferation and cell signaling, which provides a selective advantage in tumorigenesis. And it is overexpressed in malignancies, in different types of malignancies. Mostly you also find them in prostate cancers. MOS gene associated with disease, which includes oocyte, zygote, and embryo cell arrest and sarcoma cell. The COT genes are important functions in the signal activation of pro-inflammatory. That is, it increases the inflammation, promoting cancer-associated inflammations in the tumor cell, leading to papillary thyroid carcinoma. The papillary thyroid carcinogens is due to this COT gene. So these are the different types of gene which induces the RAS genes to become carcinoma cells. Then you have, that was about cytoplasmic oncoproteins. Now we are going to talk about nuclear oncoproteins. So what are the nuclear oncoproteins over there? They are among the most rapidly degraded intracellular proteins. The nuclear oncoproteins are NMYC, CMYC. CMYC can be a P62 or a P64, CFOS, cytoplasmic phos, P53, and EIA. EIA is a early region adenovirus cells, abbreviated as EIA, which are thought to have a role in regulation of cell growth and differentiation, and they have been implicated in the neoplastic transformation. Neo is new, plastic is cell growth transformation, new cell growth transformation. So here we are going to talk about nuclear oncoprotein. That's the reason why NMYC. N stands for nuclear. CMYC stands for cytoplasma. Cytoplasmic leads to nuclear problems over there. Then you have a CFOS, P53, and EIA. For example, amplification of NMYC gene has been demonstrated in the neuroblastomas. This gene is correlated with advanced stages of diseases, which leads to progression, and also it has a poor prognosis. CMYC gene, CMYC, is activated by translocation or mutations in many B-cell lymphomas. They amplify and forms tumor in different types of cells. Say, for example, small cell lung cancer and breast cancer cell. Which one? CMYC. Then when you're talking about CFOS oncogene, these are the product which has been implicated in the neoplastic transformation, which is mediating the action of variety of extracellular stimuli. So it needs to have lot of extracellular cellular stimuli, which leads to cancer cell. Then normal P53 appears to function as a transformation suppressor gene, but the mutation has been identified in both, their work has been identified in both stabilizing the protein and the result in the neoplastic transformation. We are going to talk a lot about P53 over here, and also RB cells, the ones we are going to talk about, because they are going to ask that in the examination point of view. So when you're talking about nuclear oncoproteins, you're going to talk about all this, that is NMYC, CMYC, CFOS, P53, and EIA. Now, EIA, what is EIA over here? As I told you, early region 1A of human adenovirus cells, and it plays a role in the virus replication efficiency to transform certain primary cells to cancer cells. So these are the nuclear oncoproteins, and this is how the adenovirus infection occurs, that E1A or EIA infection, early adenoma virus cells. When this is infecting a normal cell over here, transcription and viral protein is getting into the viral genome, is getting included into the normal cells over here, that is getting incorporated in the host gene over here. And once it is getting incorporated in the host gene, this leads to different types of carcinoma cells, E1A cells. Okay. Proliferation control. So how is this proliferation occurring in the cancer cell, and which are the ones that is going to control the proliferation? So cell proliferation in higher eukaryotes occurs in response to environmental signals or when the cell replacement is needed. So for example, each and every cell will have some number of days to live, and they have to be replaced. So a simple example is your RBCs. They have to be replaced every 120 days. So there is an environmental signal that is going to be given to them, or internal signal that is going to be given to them, that this has to die and a new cell has to be replaced. But some cells undergo continuous proliferation and differentiation. So growth regulatory factors generally act as the specific stages of cell cycle, commonly during first gap phase of cell cycle. And there is a number of different growth factors that is involved in them. So what are they? Epidermal growth factors, which controls the cell growth. Vascular endothelial growth factors, which controls the blood vessel development, because you don't need blood vessels everywhere. It has to be controlled. Platelet-derived endothelial growth factors controls the blood vessel development and cell growth. And fibroblast growth factors, which controls the cell growth. So these are the growth factors that controls the cells from proliferation. Excess proliferation has to be controlled, and these are the factors which is going to control the proliferation. What are they? EGFs, VEGFs, PDGFs, and FGFs. Where are you find GF? Remember, it is growth factors and the prefix over there. If it is E, epidermal. If it is VE, vascular endothelial. PD, platelet derived, and FGF is fibroblast, which controls the cell growth. If you can also see the signals that is present in this particular picture over there. Now, there is one more, tyrosine kinase inhibitors. What is this tyrosine kinase inhibitors? They block chemical messengers called tyrosine kinase, and tyrosine kinase helps to send growth signals in cells. So blocking them stops the cell growing and dividing. So this should click your some idea in your brain. Okay, when you're going to talk about inhibiting the cell growth from tumors, then you can inhibit tyrosine kinase. So inhibiting, that is when you have this messengers to inhibit tyrosine kinase, then you're stopping cells from growing. You're going to give signal to stop the cells from growing. So blocking them will lead to reduction in cell growing and divisions. So mTOR is a type of a protein called a kinase protein that trigger the cell growth. It makes or it produces proteins that trigger the development of new blood vessels, and this need new blood vessels in order to grow. So in some type of cancer, mTOR is switched on, which makes the cancer cells to grow, produce new blood vessels. But when you're going to prepare a blocker for this mTOR, it can stop the growth of the blood vessels. So this is how you're going to block them. Then there's one more, PI3K 3K cells. So these are a group of closely related kinase protein. So wherever you find kinase, that means the kinase can either increase the development or decrease the cell proliferation. So kinase proteins, they act like switches during that is they turn on the cells for development or turn off for from the cells to proliferation. So developing new treatment which can inhibit PI3Ks can inhibit development of new blood vessels. Then you have one more inhibitors called the BRAF. They block the BRAF proteins, and this can stop the cancer development because the BRAF protein can affect other proteins such as MEK, which makes the cells to divide and grow uncontrollably. And inhibiting MEK or MEK inhibitors are another type of cancer drugs. They work by blocking the MEK protein, which slows the growth of the cancer cells. So these are the blockers. What are the blockers? Tyrosine kinase blockers, mTORs, PI3K, and BRAF. These are the genes that can block the cells from going into cancerous cells. So different kinds of oncogenes. What are the different kinds of oncogenes that we'll be talking about, and how they came across with this oncogenes? So first thing is, we have to give credit to scientist Peyton Ross, because a surprising discovery was made by Peyton Ross through the study of the Ross sarcoma virus, which is also called as RSV, an acutely transforming retrovirus first isolated from the chicken sarcoma over 80 years ago. So 80 years ago, this RSV virus was identified in the chicken sarcoma. Studies of many other acutely transforming retrovirus from fowl, rodent, feline, and non-human primate species have led to the discovery of dozens of different retroviral oncogenes. You must be wondering, why are we talking about virus? The virus are also one of the reasons for the cancer development. So that is the reason why we are going to talk about this virus, and how we have come to know about this virus is because thanks to Peyton Ross, the scientist who discovered this Ross sarcoma virus in the chicken, and later on we found different types of virus even in the humans, which creates different types of cancers. So in every case, these retroviral oncogenes are derived from normal cellular genes captured from the genome of the host. And the viral oncogenes are responsible for the rapid tumor formation and efficient in vitro transformation activity characteristics of acutely transforming retrovirus. So we'll be talking about first of the cellular oncogenes or proto-oncogene. So here we are going to talk about different types over there. So under proto-oncogenes, which is also called as cellular oncogenes, which are found in the cells and abbreviated as CONC. CONC means cellular oncogene, corresponding with viral oncogene. That is, when the cellular oncogene is corresponding with a viral oncogene, then you're going to call it as SRC, that is cellular sarcoma, MYC, myeloblastomatosis, cytomatosis, and CMYB, blastomatosis, etc. They are not cancer. SRC, MYC, MYB is not cancer, but the term proto-oncogene is used to denote that their ability to require an potential oncogene so that can become cancer. Now, but this normal cells, what are the function of this proto-oncogene? They code for phosphorylate proteins, they influence DNA replication, they control mRNA production, and they bind GTPs. Why? When they bind GTPs, they become GDPs. If they're not binding, then that GTP is active at GTP place and they do not form GDP, and this also creates cancers over there. So since the retroviral oncogenes are mutants of normal proto-oncogenes, they provide key to open a door to understand proto-oncogene to oncogene conversion, how a proto-oncogene is going to get converted into oncogene. So what is the mechanism? We'll talk about one example, how a proto-oncogene can become an oncogene. One example we'll talk about is mechanism of activating C oncogene has been observed in example. So what is the example? So we are going to talk about C oncogene activation. The viral oncogene VMYC is carried by a chicken virus MC29. So this is in this picture, if you can see, avian myelocytomatosis virus 29, that is called as MC29. Now this is the virus that is going to carry VMYC gene over there, myelocytomatosis. MYC is myelocytomatosis. So MC29, this is going to carry the VMYC. The chicken CMYC gene can also be activated by another virus called ALV or avian leukosis virus. The CMYC, cellular MYC, which is a proto-oncogene, can be activated to become oncogene. Which one is that? Avian leukosis virus, that is ALV virus, can lead to making the proto to become oncogene. So what is happening over here? ALV as such does not carry an oncogene. So ALV is just an ALV, that is, it is just going to be proto-oncogene. But it can insert itself into the chromosome, chicken chromosome, next to CMYC. Now you have a CMYC over here, CMYC gene, which is not a oncogene, it's just a proto-oncogene. But when this is going to get attached beside the VMYC, when CMYC is going to get attached to VMYC, that is viral oncogene. Now this two together forms or changes the proto-oncogene to oncogene. That is, this initiate the result in overproduction of CMYC product, and this apparently is the initiating event in the development of chicken B cell lymphomas disease induced by ALV. So when they are separate, they are proto-oncogene, but when they come side by side to each other, maybe due to translocations or maybe due to various reasons, when they come side by side, then this two together produces a protein which leads to activation of proto-oncogene to become oncogene, that leads to B cell lymphomas or the cancers over there in the chicken. So that is an example of how the cellular proto-oncogene can become oncogene. Okay, so we'll talk about a little bit of human oncogenes. So that was about chicken oncogenes. Here, when you're talking about human oncogenes, they might ask you to talk about different oncogenes. What are the different types of oncogenes in humans? We're going to talk about all these cells when it has been asked for. So in 1964, the first human oncovirus was discovered when Epstein-Barr virus was detected in Burkitt lymphoma cells by electron microscope. Burkitt lymphoma is a lymphoma in the lymphocytes over there. Approximately 20% of all cancers are associated with infectious agents, and all the cancers are caused by oncovirus. So not only mutations, not only the UV rays, not only the carcinogens, even the virus can lead to cancers. The 7 known human oncogenic viruses are Epstein-Barr virus, that is called EBV virus, hepatitis B virus, that is called HBV virus. Then you have human papilloma virus, which is called as HPV. Human herpes virus 8 called HHV. This is also termed as KSHV virus. The Merkel's polymer virus, which is called as MCPYV. Oncogenic RNA virus, which includes hepatitis C virus, that is the HCV virus. Then you also have the HTLV virus. So here you have all these different types of virus, which is going to lead to tumorigenesis. That's the oncovirus. When these viruses are going to attack the normal virus in humans, that is going to lead to different types of viruses, of which are there the EBVs, the KSHV, the HCVs, HBV, HPV, HIV, and MCPYV viruses. So these are the different types of viruses that can lead to different types of cancers. Alterations in the C oncogene genes. How are these alterations occurring in the C oncogene? What is C oncogene? It's a cytoplasmic oncogene, which is also called as proto-oncogene. So when we are talking about proto-oncogenes are present in the normal cells. How this proto-oncogene can become oncogene? Example over here. One example we'll talk about in the humans over there. We talked about ALV virus and MC29 in the chicken. Now we are going to talk about what is that which is going to change the cellular proto-oncogene to oncogene in the humans. The most striking results so far obtained involves the translocations found in the Burkitt's lymphoma. That is a translocation between 8 and 14. What do you mean by 8 and 14? When there's a translocation, if you can see in this picture over here, you have a chromosome number 8 and you have a chromosome number 14 over there. If there is a translocation between these two non-homologous chromosomes, that leads to c-MYC activation. So, for example, almost all cases of Burkitt lymphoma are characterized by dysregulation of the c-MYC gene, which is under the control of immunoglobulin gene enhancer, that is the IgH, IgKN, IgL. So, immunoglobulin gene enhancer. So, what is happening? The MYC gene is found in AQ24, A8Q24. What do you mean by 8? 8 stands for chromosome number 8. Q stands for the long arm. Your chromosome is having 2 arms, P arm and a Q arm. P arm is a short arm, Q arm is a long arm. So on the Q arm, at 24th point over there, so there the gene which is present, MYC gene at Q24. This involves c-MYC oncogene translocation from chromosome number 8 to chromosome number 14. So when there is a translocation of chromosome number 8 to chromosome number 14 over there, that leads to a translocation over there. So expression of c-MYC gene results in the synthesis of transcription factors that increases the expression of other genes involved in aerobic glycolysis, providing necessary energy for cellular growth to occur. Aerobic glycolysis providing necessary energy for the cellular growth to occur. So the translocation of the c-MYC gene to IgH or IgK or IgL region moves the gene near immunoglobulin enhancer, which increases the expression of c-MYC gene. So when the c-MYC gene is increasing its expression, that means from proto-oncogene, they are going to change to oncogene, and once they become oncogenic, this is going to lead to different type of carcinomas, that is the Burkitt lymphomas, which is going to lead to any lymphatic carcinogenesis being occurred. That is when there is a translocation between chromosome number 8 and 14. Which one? The c-MYC gene that has been transferred from chromosome number 8 to chromosome number 14, and this leads to gene enhancers over there because of the immunoglobulin enhancers. That is one example over there. I hope you have understood over there. Any doubts over there, Mark? You have understood over there? Okay, so if there's no questions over there, I'll continue over there. So alterations in the expression of C-onc genes. So what is the alterations over there? The wealth of information uncovered by the genome sequencing projects over the past few decades have revealed that vast majority of eukaryotic genomes over the humans is transcribed into non-coding RNA species. Non-coding RNA species. What do you mean by non-coding RNA species? In the eukaryotic genome, you have n number of non-coding RNA species. That is, they are present, but they are inactive. They are not moving into coding of DNA over there. So these are called as non-coding RNA species. There are a number of non-coding RNA species. For example, microRNAs, MY. You have different types of tRNAs, mRNAs. But here, of these, microRNAs play an important role in regulating these human genes, including oncogenes and tumor suppressor genes. So the gene expression studies could help us to understand how microRNA dysregulation can lead to development of many types of cancers. So if there is any dysregulation in this non-coding miRNAs, then that leads to different types of cancers over there. And these are involved in regulating wide variety of cell processes, where all differentiation, proliferation, and apoptosis. So widespread evidence of gene expression studies demonstrates aberrant mRNA expression in a variety of tumor cells and tissues, which have established the involvement of miRNA cancers. If you can see in this picture over there, this miRNAs can lead to hematological malignancies, gastrointestinal cancers, non-gastrointestinal cancers, systemic diseases, so on and so forth. There are different types of cancers that can lead because of alteration in this non-coding miRNAs. So mechanism of activation and tumor suppressor genes. How are this activation being formed? That is the mechanism of activation of the proto-oncogenes and also the tumor suppressor genes. So proto-oncogenes, as we all know, that it is found in the healthy cells, and there are many proto-oncogenes present in the body. Each one is responsible for making a protein involved in cell growth, division, and other processes, and they work without any problem. But tumor cells are usually triggered by mutations in DNA, which accumulates, and this accumulation leads to oncogene trigger, which gains a function of proliferating excessively, leading to cancer development. Then tumor suppressor genes normally manage cell growth and prevent tumor development. Tumor suppressor gene, that is the genes which suppresses the tumor growth. They are present in normal cells, and this prevent any development of cancers. But mutations in these also can trigger loss of function that leads to cancer development. So here, when you're talking about activation, you can talk about three different types that is present in the genetic changes that can be present, which leads to activation of the genes to tumor genes over there. So what are the activations over there? The three genetic mechanisms which activate oncogenes in the human neoplasms are mutations, genetic amplifications, and chromosome rearrangement. First is mutation, which leads to activation of the cells. Gene amplification, that is excess of genes, that is a gene have been amplified in number, that also leads to cancers, activation of cancer. Chromosome rearrangement, if there is any rearrangement in the chromosome, that also leads to cancers, that is activation of the cells to cancer cells. So what are they? The first one, we'll talk about mutations. So when you're talking about mutations, the mutation activate proto-oncogenes through structural alterations in their encoded genes over there, and these alterations, which usually involve critical protein regulatory region, often leads to uncontrolled continuous activity of the mutated protein. The various types of mutations, such as base substitution, deletion, and insertion, all leads to activating the proto-oncogene. Substitution, that is, instead of one gene, that gene is substituted by another gene, or it can be a deletion. If you remember, 5p minus, the deletion on the chromosome number 5, and insertion, that is, if there's any insertions into the gene other than the normal gene, that also leads to activating the proto-oncogenes. In that, point mutation, for example, when you're talking about RAS family, point mutations are frequently detected in the RAS family of proto-oncogenes, and the majority of the RAS mutations involve codon 12. Remember codon 12, 13, 61, but mostly it is involving the codon 12 of the gene, with number involving with other regions such as 13 and 61. So when there is a mutation in codon 12, that also leads to mutation. Mostly you find that mutation in codon 12, but there are different types of mutations in codon 13 and 61, which also leads to activation of proto-oncogenes. The RAS mutations observed in human tumors bring about a reduction in the hydrolysis mechanism. Hence, this causes an unprogrammed stimulations of RAS MAP pathway, which manifests an increase in the cell division, leading to tumor growth. So that was mutation. The second one is gene amplification. When a gene is amplified in number, if you can see in this picture, NMYC has been amplified over there. Amplification can also lead to activation of proto-oncogenes. The process of gene amplification occurs through redundant replication of genomic DNA, often giving rise to karyotypic abnormalities. So that the karyotype is completely changed, that leads to different types of abnormalities. Amplification leads to the increased expression of genes, which in turn can confer a selective advantage for the cell growth. Amplification is increase. Now this leads to increased expression of genes, because one particular simple gene is going to give some amount, a limited amount, but when they are amplified, you're going to get excess of that particular expression of the gene proteins, which can have an advantage for cell growth. So for example, over here, amplification of MYC correlates strongly with advanced tumor cells in neuroblastoma, the nerve cells being leading to blastoma or the cancer cell, suggesting a role for this gene in tumor progression. Amplification, another example is amplification of ERB B, the epidermal growth factor receptors, which is found in up to 50% of glioblastomas and 20% of squamous carcinomas in the head and the neck region. That is amplification of ERB B genes. And approximately 15 to 30% of breast and ovarian cancers is found in amplification of ERB B2. That is another name for this ERB B2 is HER2 or new genes, which can lead to ovarian or breast cancer. All this is because of the gene amplification. Then the next one is chromosomal rearrangement. Now, this is also one more mechanism where the proto-oncogene can become oncogene. Chromosomal rearrangement over here, what is happening over here? For example, in this picture, if you see translocation between chromosome number 8 and 14. So what is happening over here? Here, the Burkitt's lymphoma, for example, the MYC gene, when it is going to get positioned at the chromosome number 14 at IgG, that leads to different types of carcinoma. That is, chromosomal rearrangement has occurred over there due to various mechanisms. That is, two different types of mechanism can occur for the translocation to take place. This translocation of the genes to different place can occur due to transcriptional activation of proto-oncogene and creation of fusion genes. Two types can occur during translocation: transcriptional activation of proto-oncogene and creation of fusion gene. What is this transcription activation? This is sometimes referred to as gene activation, which causes deregulation of proto-oncogene expression, which can then lead to neoplastic transformations of the cell. Neoplastic transformation, neoplastic cell growth, transformation of the cells. So the example over here, T814, that is translocation between chromosome 8 and 14, Q24 and Q32 translocation. Q24 from the 8 and to Q32 of chromosome 14. It was found in about 85% of the cases of Burkitt lymphoma, and this is a well-characterized example of transcriptional activation of proto-oncogene. So that was one type of mechanism for chromosomal rearrangement. Now we'll talk about fusion gene. How does a fusion genes occur? Now this is a hybrid gene formed from two previously independent genes, and this can occur as a result of translocation, interstitial deletions, and chromosomal inversion. So inversion also can play a part. Fusion genes have been found to be prevalent in all the main types of human neoplasia. So here you can see the gene transfer occurring. Here also you can see how the gene transfer occurred. The chromosome 2 has completely changed its gene over there. Protein expression will be changed over there. That is, from chromosome number 1, a gene has been transferred to chromosome number 2. So this chromosome number 2 has an extra chromosome over there. The example over here is Philadelphia chromosome, which causes chronic myelogenous leukemia, that is CML, chronic myelogenous leukemia. The translocation between chromosome number 9 and 22 at Q31 and Q11, translocation in CML fuses the CABL. So you have a CABL over there on chromosome number 9, normally located at 9Q34, with a BRC gene at chromosome number 22, Q11. Q is the long arm, Q over there. So a translocation of ABL to BRC gene over there leads to Philadelphia syndrome, which leads to leukemia. So these are the two transitional and fusion genes which causes the chromosomal rearrangements. Now coming to tumor suppressor genes. So what is tumor suppressor genes? Critical cell proliferation, including apoptosis and cell cycle differentiation, signal transduction, cell adhesion, maintenance of genomic integrity, and DNA damage repair are regulated by tumor suppressor genes. So all these are genes that has to be controlled, and tumor suppressors are going to control all these processes. Thereby, it does not allow the cancer to develop over there. So it has been generally accepted that tumor suppressor genes responsible for familial cancer syndromes can be divided into two major categories known as caretakers and gatekeepers. The tumor suppressor cells has been given the name caretakers and gatekeepers. Inactivation of both these genes contributes directly to cancer development and progression. What do you mean by caretaker and gatekeepers? We'll wind up after this. I'll just talk about what is this caretaker genes and gatekeeper genes, and then we'll wind up our class over here. So what do you mean by caretaker genes? So these are involved in the maintenance of genomic stability, and this includes gene implicated in DNA repair. So wherever there is DNA repair that has to occur, caretaker genes takes care of DNA repair mechanism. Now gatekeepers over here, what is their work over there? Gatekeepers are those genes which directly regulate tumor growth, codifying the proteins which are either going to stimulate or inhibit proliferation, differentiation, and apoptosis. So they are going to take care of how when the cell have to proliferate, when they have to differentiate, and when they have to undergo apoptosis is all taken care of gatekeepers. Caretaker is DNA repairs. Gatekeepers is they are going to take care of proliferation of the cells and differentiation or apoptosis over there. And the major genes which includes the gatekeepers and caretakers are APCs, RB1, CDKN2A, TP53, and BRCA genes. So these are the important genes that we'll be talking about under caretakers and gatekeepers over here. You will be asked to talk about RB1 and TP53 because these are the two major genes which is working for tumor suppressor genes other than the APCs, CDKs, and BRCA. RB1 and P53 genes are very important, and you will be asked these two genes in the examination point of view. So you will be talking about these in a major question, or sometimes if they're going to ask separately, we can talk about in the minor questions over there. So we'll talk about the caretakers and the gatekeepers in detail in the next class. I hope you have understood what we are going to talk about over here. That is about the tumors we are going to talk. If you have any questions, you can ask the questions, or else we will continue this class in the next class, that is tomorrow we'll be meeting. Yes, children, any doubts over here? So, okay, so in that case, we will continue this class tomorrow. I hope you have understood whatever we have done till now. Okay. So, see you tomorrow.
Okay, madam. Thank you.
Okay, ma'am. Thank you.